Good morning, students. The next topic it is molecular diagnostics. So what is meant by molecular diagnostics? It is a technique used to diagnose and monitor disease, detect risk, and decide which therapies will work best for individual patients. Okay, so what is molecular diagnostic means? It's a technique used to diagnose and monitor disease, detect risk, and decide which therapies will work best for individual patients. The treatments of a disease be effective. If you are doing for any disease, the early diagnosis is very important. Clear? So by using this method, molecular diagnostics, what is possible? Early diagnosis and understanding pathophysiology is very important. Two things. Okay, nowadays, you know this COVID-19, people are doing, I'm coming to that point next, PCR, that is called polymerase chain reaction, because we have to diagnose little early. Okay, so that is called early diagnosis. The next conventional method of diagnosis, it was serum and urine analysis. Early detection of disease is not possible. Clear? So conventional method, whichever the conventional method of diagnosis, serum analysis or urine analysis, but in that case, early detection of disease, it is not possible. So how we can do the early diagnosis? It is possible with advanced molecular diagnostics like recombinant DNA technology, PCR that I told. Nowadays it's very common, you can read in even the newspaper, PCR test. For this COVID-19, we can read PCR test. What is meant by PCR test? Polymerase chain reaction. It means that is early detection or diagnosis of the pathogen or virus or whatever it may be. Okay, so we can find out very in early stage. So one is recombinant DNA technology, second is PCR or polymerase chain reaction, third is ELISA. What is the full form of ELISA? Enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. So these are the methods we used to find in early stage. Okay. So these are examples for molecular diagnostics. I told early diagnostics is possible with advanced molecular diagnostics. So which are the advanced molecular diagnostics? One is recombinant DNA technology. Second one is PCR or polymerase chain reaction. Excuse me. Third one is ELISA or enzyme linked immunosorbent assay. So conventional methods detect presence of pathogens like bacteria, virus, etc. When disease is well established and produced disease symptoms. So it means by that time pathogen is in high content in the blood. So that is how conventional methods detect the presence of pathogen. Which are the pathogens like bacteria, virus, etc. When disease is well established. That was a conventional method. When disease is well established and produced disease symptoms. Okay, so by the time pathogen is in high level in our blood. Okay, what are the significance of molecular diagnostics? What are the importance or significance of molecular diagnostics? Molecular diagnostics like PCR detects pathogens even at very low concentration. So conventional method we told we can find out only when the pathogen is very high quantity in the blood or more quantity in the blood. But here molecular diagnostic like PCR detects pathogens even at very low concentration or amplification of nucleic acids of pathogens. Second point, what are the another significance or importance of molecular diagnostics? To detect HIV in suspected AIDS patients. So we are using this molecular diagnostic methods to detect HIV, human immunodeficiency virus, in suspected AIDS patients. Third one, to detect mutation in genes in suspected cancer patients. For that also we are using molecular diagnostics method. In early stages we can identify it. To detect mutations in genes in the suspected cancer patients. Then next significance, to detect many genetic disorders. So detect many genetic disorders, we are using this molecular diagnostics method. Then PCR, we told what is meant by PCR, polymerase chain reaction. PCR requires single-stranded DNA or RNA tagged with radioactive molecule to hybridize complementary DNA in clone. So what it requires? Single-stranded DNA or RNA tagged with radioactive molecule to hybridize complementary DNA in clone. The next, following hybridization, the next step it is autoradiography. So what is meant by autoradiography? It means a technique using X-ray film to visualize molecules or fragments of molecules that have been radioactively labeled. Autography has many applications in the laboratory. So after this hybridization, next step what it is? Autoradiography. So I'll tell you what is meant by autoradiography. A technique using X-ray film to visualize molecules or fragments of molecules that have been radioactively labeled. Autoradiography has many applications in the laboratory. The next, clone with mutation or mutated gene not appear in photographic film. So here the probe will not have complementarity with mutated gene. So what is meant by genetic probe? Genetic probe is a fragment of DNA or RNA of variable length, usually 100 to 1000 bases long, which is radioactively labeled using DNA or RNA samples to detect the presence of nucleotide sequences that are complementary to the sequence in the probe. So that is the meaning of genetic probes. So I'll tell what is meant by genetic probes. Genetic probes is a fragment of DNA or RNA of variable length, maybe usually 100 to 1000 bases long, which is radioactively labeled, used in DNA or RNA samples to detect the presence of nucleotide sequences that are complementary to the sequence in the probe. Clear? The next, what is the principle of PCR? You can see here. First one is clinical sample. Second, DNA preparation. Third, amplified target DNA plus probes. It gives detection system. So that is the principle of PCR, polymerase chain reaction. The next, before we told one term ELISA. ELISA means another molecular diagnostics to detect antigen. What is meant by antigen and antibody? Suppose one virus is entering into the body, that virus is called antigen, and your body will produce some substance to kill that antigen. They are called antibody. Clear? So ELISA, another molecular diagnostics to detect antigen, or we can tell foreign substance or pathogen. That is called antigen. Maybe the substance which entered from outside, that is foreign substance or pathogen. That is called antigen. This ELISA works on principle of antigen-antibody interaction. So how it will detect? This ELISA detect pathogen by presence of antigens like proteins or glycoproteins, etc., or by antibodies synthesized against pathogens. So how it is detecting? In the early stages, ELISA detects this pathogen by presence of antigens. I told in early stages very low concentration, but in late stages it will become more content in the blood, concentration will be more. So ELISA, this method, these molecular diagnostic methods we are using always to find in the early stages, even PCR, all the methods. So here it detects pathogens by presence of antigens like proteins or glycoprotein, etc., or by antibodies synthesized against pathogens.